Hello, everyone. My name is Jiuk. Please call me G. And it's my great pleasure today to share my work today at the PCTC meeting. As a member of the Zahn Lab, I study developmental hematopoiesis in zebrafish. And today, I'll talk about my project, which looks at how GCSF mobilizes hematopoietic stem and progenitor cells from the embryonic hematopoietic niche. So mobilization is a process in which certain drugs are used to release hematopoietic stem and progenitor cells, or HSPCs in short, from the bone marrow into circulation. And GCSF is one of the most widely used mobilizing agent in clinic. It is used during allogenic stem cell transplants, for example, to collect stem cells from the donors. And it's also used during autologous stem cell transplants to collect stem cells from patients. Now, in both cases, hematopoietic stem cells are mobilized from the adult hematopoietic niche into circulation. Now, a lot of studies suggest that GCSF-induced mobilization of stem cells is not entirely cell autonomous. So in other words, niche cells, or the components of the stem cell microenvironment, takes part in mobilizing stem cells from the bone marrow. So for example, GCSF is known to expand and activate neutrophils. Activated neutrophils then secrete matrix metalloproteinases into the extracellular matrix, which creates a very proteolytic microenvironment. Now, in this proteolytic environment, proteins that hold stem cells in the bone marrow, such as the CXCL12-CXCR4 axis, is disrupted, and hence stem cells are mobilized into circulation. Now, other studies also suggest that GCSF reduces number of osteoblasts in the bone marrow, which are crucial for supporting stem cells in the bone marrow. So all of these evidence suggest that the bone marrow niche plays a critical role in mobilizing stem cells. Now, stem cell and niche interaction in the adult hematopoietic niche is also found and conserved in embryonic hematopoietic niche. So for mammals, fetal liver serve as an embryonic hematopoietic niche where stem cells can stay and expand until birth. Now, structure-wise, stem cells reside near fetal liver endothelial cells and stromal cells, like they do in the bone marrow. In terms of signaling pathways, cytokines such as CXCL12 or KIT ligand maintain the hematopoietic stem cells in the fetal liver as they do in the bone marrow. So given all these similarities between the adult and embryonic hematopoietic niche, I came to ask myself, can GCSF also mobilize HSPCs from the embryonic hematopoietic niche? And if so, how does it do it? Now, zebrafish is a wonderful model organism to study developmental hematopoiesis, especially since I'm a member of the Zahn Lab. And first of all, zebrafish has both embryonic and adult hematopoietic niche, just like the mouse and also the human. For zebrafish, during 2 to 5 days post-fertilization, when fish is still considered an embryo, hematopoietic stem cells resides in the caudal hematopoietic tissue, or the CHT. Now, CHT contains endothelial cells and stromal cells in which HSPCs can reside in, just like they do in the mammalian fetal liver. So it's comparable to fetal liver. Now, after 5 days post-fertilization, stem cells move to the kidney marrow and the thymus, which are equivalent to mammalian bone marrow and thymus, the adult hematopoietic niche. Also, in zebrafish, we can easily visualize and see where stem cells are at a given time point. We can live image a zebrafish to examine whether stem cells are in the niche or in circulation. And here, I'm showing you time-lapse movie of HSPCs in circulation in a live zebrafish. We can also perform whole mount in situ hybridization to get a snapshot information of where stem cells are. And here you can see that HSPCs are in the CHT at day 4. But then at day 5, we can start seeing HSPCs in the kidney, marked here, the adult hematopoietic niche. So we can easily visualize where stem cells are at a given time point. Hence, zebrafish is a wonderful model organism to tackle my question and study developmental hematopoiesis. So in zebrafish vocabulary, the overarching question is this: can GCSF mobilize HSPCs from the CHT? So to address my question, I first established an inducible way of expressing GCSF in zebrafish embryos. I cloned GCSF under a heat-inducible promoter, as you can see here, which I then delivered into one-cell stage embryo. And at day 3, when HSPCs are in the CHT, the embryonic niche, I gave a heat shock to induce expression of GCSF. Now, GCSF is known to increase the number of neutrophils. Therefore, to validate whether GCSF is successively being induced in this system, I looked at a transgenic line with green neutrophils and tried to assess whether I see any increase in number of neutrophils. And indeed, 24 hours after induction at day 4, I observed almost 6-fold increase in number of neutrophils, validating that this system is indeed working, that GCSF is being expressed when I give the heat shock. So from now on, I'll be using the same scheme to express GCSF in zebrafish. Heat shock at day 3 when stem cells are in the CHT, and then assess any phenotype at day 4. So having established the inducible GCSF system, I started off by imaging HSPCs in zebrafish embryos, particularly the CHT. More specifically, I imaged Rungsem cherry line, the reporter line, where HSPCs are labeled red. And the very first thing I noticed was a notable increase in circulating HSPCs. And here I contoured the blood vessels with white lines, as you can see here. And on the right panel, when GCSF was expressed, there are more circulating HSPCs, as you can see here. This is exactly what I'd expect to see if HSPCs are being mobilized from the niche and enter circulation. So this was a very exciting observation for me. Now, another observation was an accumulation of HSPCs at the kidney marrow, the adult niche of zebrafish. Again, I used a red stem cell line to look at the kidney at day 4. Now, normally at day 4, HSPCs will not be at the kidney like you see here because HSPCs are still stuck in the CHT. When GCSF was expressed, however, I saw HSPCs that have prematurely colonized the kidney, as you can see in this bottom panel. Now, this was also confirmed with a whole mount in situ hybridization for HSPCs. In nearly 70% of 4 days old embryos where GCSF was induced, I observed HSPCs that prematurely colonized the kidney, like you see at this bottom panel. So these data suggest that in response to GCSF, HSPCs mobilized from the CHT into circulation. And it also looks like some of these mobilized HSPCs that are now in circulation reaches the kidney and prematurely colonizes the kidney, which you'd otherwise not see in wild-type embryos. Then I started asking myself, well, is this simply because the total number of HSPCs have increased? Did GCSF just induce proliferation of HSPCs, and therefore am I just seeing more HSPCs everywhere, more stem cells in circulation and more stem cells in the kidney? So to address this question, I performed EDU proliferation assay to quantify number of proliferating HSPCs. And when I calculated the proportion of EDU positive HSPCs, it turned out that there is no significant increase in proliferating HSPCs in response to GCSF. Also, I simply counted the number of HSPCs in the CHT using 2 different transgenic lines that labels HSPCs. Here I'm showing images of just one transgenic line, but I did quantify both lines, as you can see on the right. And it turns out that GCSF does not increase, if not decrease, the number of HSPCs in the CHT. In fact, this decreasing tendency you see here supports the idea that GCSF is releasing and mobilizing stem cells from the CHT into circulation. So this, along with the EDU data, suggests that the total number of HSPCs does not increase in response to GCSF. Therefore, increase in circulating HSPCs in response to GCSF is most likely attributable to the mobilization of stem cells from the CHT. So lastly, to get some mechanistic insight, I asked myself if neutrophil is anyhow involved in this mobilization. And a simple way of testing this is by getting rid of neutrophils and see if mobilization still proceeds. So I employed a chemical neutrophil ablation system on top of the inducible GCSF system. Here I have a fish with neutrophils expressing nitroreductase, and at day 3, I added a drug called nifurpyranol, which reacts with nitroreductase to kill the neutrophils. Now, 24 hours after drug treatment, the nifurpyranol treatment, I assessed whether the number of neutrophils decreased in this system. And here in this image, you can see that this system effectively gets rid of most neutrophils in zebrafish embryos, labeled red here. And also from quantification, you can see that there's a stark decrease in number of neutrophils using this system. So the ablation of neutrophils seems to be working. And so now, to assess whether neutrophil is necessary for GCSF-induced mobilization from the embryonic niche, I performed in situ hybridization in neutrophil-ablated embryos to see if there are any premature kidney colonization of HSPCs. And if neutrophil is necessary for the mobilization, then stem cells will not prematurely colonize the kidney at day 4 when neutrophils are ablated. Now, here's the result, and I'd like to draw your attention to the 2 panels on the right. Here we can see that HSPCs prematurely colonize the kidney, regardless of the neutrophil ablation. And the proportion of embryos with the premature colonization of the kidney was not significantly different. Now, this suggests that neutrophil is not necessary for GCSF-induced HSPC mobilization from the embryonic niche. It's highly likely that there is a neutrophil-independent mechanism that mobilizes stem cells from the CHT. So to sum up, I started off by asking whether GCSF can mobilize HSPCs from the embryonic hematopoietic niche. And I did find out that yes, GCSF mobilizes HSPCs from the embryonic niche, the CHT. But it seems like neutrophil is not necessary for this mobilization. It seems like there will be a neutrophil-independent mechanism on how GCSF mobilizes stem cells from the embryonic niche. Now, in the future, I'd like to continue my search of how GCSF mobilizes HSPCs from the CHT. And in addition, I believe that this project lays a foundation for using zebrafish embryonic niche to study mobilization mechanisms, since it's very easy to visualize the stem cells and also the compartments of the niche. So with that, thank you for listening, everyone. I'd like to thank my lab for their support, especially Len, who gave me precious advice on driving this project forward, and also to all the lab mates for their wonderful academic and mental support in the lab. So thank you very much, and I'd be happy to take any questions.